Hello? Can you hear me? So first, I would like to apologize. We are a bit late, and that's due to the never working trains. And due to the never working trains, there was not any possibility to get a cab here. So our nice speaker need to take a long walk. That's why we are a bit late. We are really happy to have all of you here, but we are extremely happy to have the presenter here, of course. And today is Jerker Widengren. Oh, I need to tell you who I am. I'm Sofia Huber, and I'm chairperson for this, the department, or that actually are hosting this seminar today. Sorry? Speak up. Högre. I don't know. Can you hear me now? So, Jerker Widengren, professor of KTH, he is the one that had assembled this nice seminar today. And we are very happy to have Professor Michael Orit here, as well as Professor Xiaoweng Shuang, that will give us a very interesting talk. And also Jerker Widengren will talk somewhat about his research, and then we will hear Johan Strömqvist, who is heading a company that using this kind of technology. And I will not talk anymore today. I will give the word to Jerker, who will actually be hosting this and taking care of everything and keeping track of the time as well. Thank you.
Thank you. I try to do my best. And sorry about this delay. I thought I will give a short introduction before leaving the word over to the main speakers, giving this topic, putting it into a biological and somewhat historical context as well. And this is how I usually start my lectures in the biophysics course at KTH, given to engineering students, really to point out that biology is complicated. And if you go down on the molecular scale, we have the proteins, and they perform a large variety of tasks. And for that reason, they also show a tremendous variability. So if you look on the amino acid sequence of a protein, I mean, for each amino acid, you have 20 choices, and then for a protein with, say, 100 amino acids, you can think of the number of possible combinations of the sequence of amino acid, just that. But this is, of course, not the full story. It's just a small explanation of the function of a protein. Then this sequence, this chain of amino acids can form into a structure, three-dimensional structure, and there is some kind of structural function relationship. But this is also not the full story. You also have the dynamics of the protein. You have to consider also the environment in which the protein is working, as well as the other proteins or biomolecules with which the proteins are interacting. And then going from the nanometer scale up to the scale of micrometers, we arrive at the scale of the cells. And there we see that we also have a tremendous complexity in the biological system, with lots of different pathways and regulation mechanisms, as well as different organelles with specific functions. So taking that further, we see actually at every level of biology, we are faced with a tremendous complexity. And so we need very strong, powerful tools actually to address and try to answer the underlying mechanism in order to understand biology. And one such tool or a category of tool is spectroscopy. That is the study of how electromagnetic radiation interacts with matter. And then we have fluorescence spectroscopy. So you can basically do spectroscopy over the full electromagnetic spectrum, and then fluorescence you do in the range of UV light, visible light, and infrared light typically. So what you do, you excite fluorescent molecules with light, you generate an electronic excitation, and upon deactivation of that excitation, you have the emission of fluorescence. But during this excitation, the excitation cycle, you lose some energy such that the emitted fluorescence is redshifted. And that makes it possible actually to spectrally separate the emitted fluorescence light from the excitation light. And that was actually done already in the mid-19th century by George Gabriel Stokes. He used at that time quite simple instrumentation using the church window for filtering out the excitation light, proper wavelengths. And then he used a glass of white wine as an emission filter to filter out only the emission of the fluorescence from quinine, which is a substance that, well, it's actually a malaria drug, and it's also found in tonic water, for instance. So what is special about fluorescence is that you can achieve enormous sensitivity down to the single molecule level by this spectral filtering, on the one hand, but also by suppression of background by other means, and by having also a high signal level. You can generate high photon fluxes of fluorescence photons. And adding to that, you also have a specificity in many ways. One is that you can characterize the fluorescence light by many parameters, like not only intensity, but also wavelengths, polarization, and fluorescence lifetime. So looking a little bit on the history of single molecule detection, it all started, well, it's a matter of definition, of course, but one can take 1960 as a starting point. Then there was a paper published by Boris Rothman showing that you can detect the presence of single enzyme molecules by having a substrate, a fluorophore labeled with a sugar part, which is non-fluorescent, and then with the enzyme acting on that, cleaving off the sugar, you get a fluorescent product. And then by putting enzymes, a small number of them, individual ones, 1 or 2, in small vesicles in an agarose gel, and then observing the fluorescence in the different vesicles, that could be taken as a signature of single enzyme molecules. So in an indirect way, but still single molecule detection by fluorescence. And then moving to the next decade, we had the report by Thomas Hirschfeld using so-called total internal reflection microscopy, looking on individual proteins on a glass surface and showing that it's possible to detect them. Now, with putting lots of fluorophore marker molecules on each protein, hundreds of them. But by having hundreds of fluorophore marker molecules on one protein, he could show it is possible to detect directly individual molecules. And what he also pointed out, which I think is kind of seeing the potential in it already at this time, is that single molecule detection by fluorescence can be combined with the use of antibodies to reach a high specificity in where the antibodies then bind to a molecule of interest to visualize, which is actually quite well ahead of time in ideas of that kind. And then, one decade later, we arrive at 89 and 90. Then, for the first time, it was shown that one can also detect single fluorophore molecules, not hundreds of them per molecule. And that was done by Mörner and Cador by absorption, and then at about the same time by Michel Aurit and his colleague Bernard by the use of fluorescence, which actually has proven to be the method of choice to analyze single molecules. And this kind of set the ground for a rather quick development thereafter, showing that also it's possible to detect single molecules in aqueous solution. These detections in 89-90 were done at cryotemperatures in organic crystals. But it's also possible to do in aqueous solution and at room temperature. And that, of course, then opened up for the biological applications of single molecule detection. I also have to point out at this stage, this also put the prerequisite for the development of fluorescence correlation spectroscopy. So the ability to detect individual or a low number of molecules makes it possible also to look on stochastic fluctuations from a few number of molecules, which you cannot do on an ensemble level. And that opened up the possibility to look on the dynamics of molecules. And in that development, of course, I also want to mention Rudolf Riegler sitting in the back, and my supervisor at the time. And I was fortunate to get into this and contribute to this development at around 1990 under the supervision of Rudolf. So one can say that these days detection of single molecules by fluorescence is not an issue. It's more how one can use it. And I think in biology that there are numerous examples. So you can look into dynamics under thermodynamic equilibrium, stochastic fluctuations, and you can look into heterogeneities that are hidden on an ensemble level when you look on an enormous number of molecules at the same time. But there's one more, well, there are several, but there's one milestone also in the development of single molecule detection by fluorescence. And that is the use of single molecule detection by fluorescence to achieve super resolution microscopy. And this was done by Xiaowei Zhuang and Erik Betzig at 2006, 2007 about. And the idea is that you cannot optically resolve the image of a single molecule because of the diffraction limit. That will limit the resolution. So you will see an image of a single molecule like a point spread function, a blur. But what you can do with high precision accuracy is that you can determine the maximum of this image of the single emitter. And if you then detect a sparse number of emitters well separated from each other in one image, and then switch them off, detect another set of single emitters, and then add the whole stack of images of single emitters, you can then generate an image with super resolution, down to tens and even below nanometers of resolution. So to conclude, fluorescence spectroscopy, the ability to detect single molecules, opens very interesting perspectives in biology. And it's my true pleasure, and I'm very glad to have two of the real main profiles in this area here at this symposium, Michel Orit and Xiaowei Zhuang. And it's not only that they made very important footprints in the history of single molecule spectroscopy, they have continued to do very interesting research, and I think you will be convinced about that this evening as well. So with that, I leave over the word to
While we're setting up, I think the microphone is working. Yes, you can hear me. Huh? Can you hear me clearly? Yes, thank you. All right, so first of all, let me thank you, Jaka, and congratulate you with your election at the Royal Academy of Engineering Science. It's a very nice event, also a nice opportunity that we get to see each other again after a few months. And while the computers getting set up, let me tell you a few words what we do in our group. So our group is in Leiden, Holland, and we are doing experiments with, that's not this one, it's going to switch. We are doing experiments with single small objects, molecules or nanoparticles, sometimes quantum dots, in a normal optical microscope. This is the current composition of the group. Ah, that's not the right, yeah, so you have to go back. Yeah, that works. Yeah, can you see the pointer? I don't see it very well. It's difficult to see. You see it? Can you see the pointer? Yeah, it's a bit weak. Yeah. Yeah. All right. I will try to put. It's very, very weak. All right. Okay. I will not show the outline because I've decided to shorten the talk a little bit. Let me explain to you briefly how we detect a single molecule. The idea is to have an optical microscope, and as Jaka just told us, we, of course, are limited by diffraction, by the Hubble resolution of light, which means that the spot of light, the best spot of light we can. Oh, thank you very much. The screen is very bright. Yeah. Yeah, so I think it doesn't help. So the best spot of light we can make is typically a few hundred nanometer in size, and of course it is way larger than a single molecule. So there are two consequences. The first one, this limits the accuracy at which we can pinpoint the molecule. There are ways around that. And the other problem is, namely that there are a very large number of other molecules in the focal spot that we have other than the molecule we want to detect, right? And this is basically, if we take a very small spot, the kind of order of magnitude that we have is 10 to the 9 molecules, 1 billion molecules in the focal spot. So the problem is to find one molecule out of 1 billion. It's a little bit like finding one person in the whole of China. It's a very difficult problem because simply the number is too large. There are not so many physical techniques that allow us to find a signal of 1 in 10 to the 9. You can think of many techniques in physics, infrared absorption, or all kinds of signals. They have backgrounds. And you need a technique that is able to give you a 1 in 10 to the 9. And this is possible with fluorescence. And the reason for that is that fluorescence is a resonant technique. So we are able to excite the one resonant molecule with the laser, and this signal will dominate the signal of 10 to the 9 non-resonant molecule. And the key word is resonance. The difference that the molecule that we excite is resonant with the laser, absorbs the laser light, whereas the other molecules, the protein molecules, or the water molecules, or the lipid molecules, they just do not interact, or very weakly interact with the laser. They do interact a little bit. It's called Raman scattering. So they will also give us some light shifted from the laser wavelength by more or less the same amount as the dye molecule would observe, but with an efficiency which is 10 to the 12 times weaker. And that means we can see a signal to background of 1 in 10 to the 12 with fluorescence. So that's the key of the whole talk and of actually the introduction of Jerker. Fluorescence allows us to select one object in 10 to the 12 at the maximum. We don't need to go to 10 to the 12. We can need to 10 to the 9, and then already you can see one single molecule. All right, so I will not explain all the details. You can see here basically what we have in most of our experiments, an excitation laser, which has some yellow, green color, and then it produces fluorescence, which is Stokes shifted, downshifted in energy most of the time. I will show you one example where this is not happening. Not the only thing that is happening, but most of the time it's downshifted in energy, or long shifted in wavelengths, and this color is simply collected, transmitted through a filter, and detected by a single photon counting electronics. So simply a detector that gives you a click for every single photon that is detected. So with this setup in mind, what we have to do now is just dilute our sample so that at every point of a sample we have at most one molecule. So most of the time 0 molecule, that's a dark background you see everywhere, and from time to time 1 molecule. And you can see immediately these molecules, there are little spots, and the size of the spot is of course the point spread function of the microscope. It's diffraction limited. In order to go beyond this, you need to use the super resolution techniques that Jerker introduced and that Xiaowei Zhang will explain to you, at least for the storm part, in the next talk. So you see immediately that these spots that we have, they have different shapes. You see this one, for example, is very round and regular, very smooth. That's not a single molecule. It's actually a piece of dust that has gathered maybe 100 molecules from the intensity. The other spots are much noisier, and that's what we call blinking, and it's a very interesting phenomenon. On the one hand, it's, of course, a limitation in the signal. While the molecule is blinking, it's dark, you lose information. On the other hand, you can monitor this blinking, and we see that the intensity, although the laser is completely constant in time, the intensity of fluorescence is switching between on level and off level in one step. It goes on, off, like a random telegraph. And this very characteristic signature is something that tells us this is a single molecule. So if we, or at least a single quantum system, a single emitter, if we did have 2 or more of these emitters in the same spot, then we have intermediate levels. It could be 2 emitters off, 1 on, the other off, or 2 off. So you have multiple levels. If you see 2 levels, you know it's a single emitter, right? So this is one of the ways in which you can use this information to obtain super resolution is to exploit this blinking parameter. So let's now try to see some examples of how to exploit this. As an example, I will take just a very simple example, rotational diffusion of a dye molecule in a liquid, glycerol. So glycerol has this property that when you cool it down, it doesn't crystallize very readily. It becomes more and more viscous, at first like oil, and then like tar. And then if you go lower and lower, if you go to temperatures of about 200 Kelvin, this is not very cold, but nevertheless at this temperature, glycerol becomes completely solid, like a glass. It's as hard as glass. And that means that if we put molecules in this medium, they will move more and more slowly as we decrease the temperature. So we can start at room temperature, where the rotational time of this dye molecule, for example, this PDI molecule, will be on the order of microseconds. If we cool down, we go all the way to seconds. So we win something like 6 to 9 orders of magnitude in time. And this allows us to do very easy experiments because the rotation time is so long that we can measure easily with many fluorescence photons. So we started these experiments a long time ago. And this is just an example of data where you see 3 different individual molecules, and you see the pixels, because we are scanning the laser, and you see also color. And the color just here indicates the direction, the polarization of light. So if the color is red, it means the light is vertical. So the molecule is, by chance at this moment, oriented along the vertical axis. And if you see green, it means horizontal. The molecule has rotated and is now emitting horizontally. And you see that different molecules move in different ways. You see this one, of course, red, 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 and becomes green, green, green. It's moving slowly. The upper one appears to be switching much faster. It's first red and green and red and green and red and green again. So it's really moving much faster than the first one. So that's very qualitative at this point. So can we do that a bit more quantitatively? Yes, we do, by correlation spectroscopy, a technique that has been pioneered and used a lot by Rudolf Riegler in Stockholm. So what we do here, we just record vertical and horizontal traces of the fluorescence, and we do a cross-correlation, or we just measure the linear dichroism and correlate it with itself. And with this, we get the time it takes for the molecule to change orientation. And if you do that, we have a nice illustration of what Jakob just told us. When you look at single molecules, you reveal the disorder, the heterogeneity, which is hidden in the ensemble measurement. If you do an ensemble measurement, you get the gray line that is difficult to see probably in the middle of this cloud of points. So this is time as a function of temperature. So when we change temperature, you see the ensemble measurement has this boring, linear, almost linear dependence. If you measure molecules at the lowest temperature, you find a cloud of points. So all these molecules move at different rates. And these rates are really real quantities. This is not noise. This is thing that you can really measure several times after one another. And not only that, you can also change the temperature. Here we are increasing the temperature from 205, 207, up to 212. And you see that the lines here are joining the points correspond to the same molecule. So the same molecule which diffuses slowly, has the longest time, remains slowly when you change temperature. This experiment is very long. It takes 1 day approximately to go through all these temperatures. But still a molecule which diffuses in about 1 second remembers during the whole day, from the breakfast in the morning until you go to bed in the evening, it remembers I have to diffuse very slowly. And the one which diffuses fast also remembers I have to diffuse fast. There are a few changes, but not that many. So that's very surprising, and that goes a bit against the conventional wisdom in the field of glass theory, where people thought, well, glass is a bit like a liquid in slow motion. This is not at all the case. You find here, by putting single molecules in different areas of the material, you find different local dynamics, different local viscosities. Yeah, that's a surprise. And there are interpretations with that. I will not go into the details. This is an experiment by Dave Waits in Harvard on colloidal glasses, where you see here this mobility lake. So it's easy to assume now that if our molecule is in that lake where these particles are moving in correlation, or in that other lake, the blue one, where a different set of particles are moving, of course the local viscosity will be different. Those are completely different areas that have nothing to do with one another. And the purple dots or the blue dots here are the particles which do not move, or do not move at least on the measurement time of our experiment. And this make these fixed molecules are a little bit like solid walls, like in this room. We can move inside, and our neighbors can also move, but we can dance here and they can sleep on the other, or try to sleep in the other room, without any interference between the two, because we have solid walls which don't move. So this is basically the picture that the single molecule measurements suggest to us. All right, now I'm switching to a different subject. Recently, we studied in collaboration with my colleague Jan Kantas and Thijs Aartsma this protein, copper azurin. They have a long experience of this protein. They know how to do it, and we started to do single molecule measurements. So this has been done essentially by this excellent PhD student who became a young doctor last month, Biswajit Pradhan. And he has done this single molecule measurement on azurin. And in order to measure this protein is in charge of electron transport in the cell, in bacterial cells. And in order to take electron transport, it has a copper atom that can be reduced. And then of course, it takes an electron, reduces the copper, and the copper is white in this case. Copper salt has no significant absorption, invisible. And then the protein goes to some other place, gives the electron to another partner, and in this way, the copper gets oxidized. It becomes copper 2 plus. Copper 2 plus is colored. Think of copper sulfate, it's blue. So copper 2 plus absorbs in the green, in the blue, green, yeah. In the red, sorry, so it has a green color. So what we do now, we'll make this, take this protein, attach a dye in the vicinity of the copper, and when the copper is reduced, the dye will fluoresce, give us photons. When the copper will be oxidized, because of the absorption of the copper in the red, it will be possible to accept, for this copper, to accept energy from the dye and to quench the fluorescence. So by monitoring the intensity of the fluorescence, we know whether the copper is oxidized or reduced. So the next step that Biswajit did is build this cell, so in which you have a control of the electrochemical potential under the microscope, so we can at the same time monitor the fluorescence of the molecule and control the redox potential by changing the voltages on the potential stat. And you see here, for example, if you go to oxidizing potential, the immobilized molecules here are not fluorescing except for a few ones, which probably are dead or they have no copper, they lost their copper. And when we reduce, a lot of these molecules brighten up and appear. So now when this becomes interesting, then when we record time traces, and we can do the same thing not in static conditions, but as a function of time. And typically you see that under reducing potential, we have long on times with a few interruptions, the off times, the bright and dark times. And when we go to oxidizing potential, that's just the opposite. We have very short on times. So the dye is almost emitting a few photons, then go to the dark state, is quenched, and then we have nothing, and we have emission again. So by studying these successions of bright and dark times, we know something about how the protein is working, right? This gives us information because the function of this protein is to transfer electrons. So we have a signal that monitors the active state of the protein. Right, so we did some statistics. I will not go into details because I think it's a bit too, it would take me too far. What you see typically, you have the signature of this non-exponential distribution of times, which have to do with dynamical heterogeneity. So we find again the same theme as with the glycerol. You will remember in the case of glycerol, we have this spatial heterogeneity. Here we're looking at the number of molecules, and we again seem to find something like spatial heterogeneity. So we have this histogram of times, which has been made in this case by collecting many different molecules. And now the question that immediately appears is this disorder, this heterogeneity, is it a signature of each molecule? Molecule A is different from molecule B, and they have a different exponential distribution. When we average all these distributions, we find a stretch exponential, a non-exponential distribution. That's explanation number one. And explanation number two is maybe even the same molecule as a function of time is changing. So at some time it has a short lifetime in the dark state or bright state, and at a later time this time becomes longer. And that's called dynamic heterogeneity, and on the temporal time or spatial time. Not time, a temporal side or spatial side. Right, so actually what we did here, we applied, this is one single molecule, and this is just an example to show you that actually this heterogeneity is already present on a single molecule level. So it's not only spatial heterogeneity from molecule to molecule, but it's time heterogeneity. So the same molecule takes different conformations with them. So Jaka told us in his introduction, biology is very complicated, the protein is very complicated. There are so many possible sequences, but even with one fixed sequence, there are many, many degrees of freedom in the flexibility of the molecule that allow for a very large number of conformations. This is called the potential energy landscape of the protein, which is a very complex surface. And what we find here is that actually, while the molecule is performing its function in our cell, it's also changing conformations. And what you see here, for example, the time trace seems to have a change, but it's very noisy, so you have to go more quantitatively. So what we have done here is look at the dark times and bright times, and we have averaged them a little bit, because if you don't average, there is more noise. So we average about 10 of them, and you see that the average of these dark times is really fluctuating a lot. The bright times and the dark times is changing a lot. We can also make the ratio of dark to bright, and this gives us the midpoint potential. That's a characteristic of the protein. You see that if you do this ratio, this midpoint potential is also moving. So now there are, of course, a lot of assumptions in the way we evaluate this data. We use a so-called change point potential, a change point algorithm to determine the time at which the fluorescence change state. So it would be nice to do something independent of any such algorithms. So we have done that as well. This is shown here. So this is even simpler. What we do here is, again, correlation function. So we take the same trace of a single copper azuline molecule, and we cut the trace. It's a long trace, about several minutes. We cut it into small pieces, and then we do for each of these pieces different analysis. For example, lifetime histogram or correlation function. And you see that this correlation function is changing a lot. Not only does the contrast of the correlation change, but also the time is changing. So it's really, and here we have no parameter. It's just averaging. So the only thing we do, we cut the trace in small pieces and do the normal averaging without any adjustable parameters of each piece independently. And you see that these things are changing with time. As a control, we also looked at a zinc azuline that does not do this electron transfer with different dye that has blinking. Of course, if the dye doesn't blink, you cannot really compare because the intensity is very constant. So that's trivial. So we have something in which the dye is now blinking because of redox reactions of the dye, and the protein is doing nothing. In this case, nothing happens. The dye is completely constant, so this lifetime is completely constant. The correlation function, which is due to the blinking, is also constant. Nothing changes. So we really think that is a strong argument to say what we were seeing before is really something which is from the protein and not from the dye. And I should stress here, of course, that we see that there are probably very, very subtle motions. Actually, azuline is very well known to be a very sturdy, very stable protein, and protein for physicists that is easy to work with. And you would not expect to have a lot of dynamics in this protein. But we are looking at the electron transfer, right? I mean, the electrons to go inside the protein, reduce the copper, or go out, oxidize the copper and go out. In both these cases, you need to transfer electrons. And electron transfer, as you know, is extremely sensitive to the details of the path for electron transfer. So even a small change is going to have a large influence on the rate of this reaction. So I think that's why, like in a work by Sonishi and Ho Yang back in 2003, they showed that in another case, by probing electron transfer between a dye molecule and a tyrosine, and an amino acid in the protein, it was possible to follow conformational changes of the protein. I think this is very similar. We probe these subtle conformational changes because we look at electron transfer, which is very sensitive. If you would look at thread, for example, it would be much more difficult. If we look at structure, it would be also much more difficult because structure is within a few angstroms, and we don't have the resolution to resolve changes of a few angstroms. But electron transfer has it. All right. So now we'd like to tell a few words about single gold nanoparticles, because we've been busy with this the last few years. And I need to introduce briefly what plasmon is for me. So that's so-called plasmonic fields. So in plasmonics, a plasmon, in principle, is an electromagnetic wave, is light that propagates at the interface between a metal and a dielectric. For example, a surface of gold and vacuum, you'll have a plasmon wave. But if you make the gold particle small enough, the plasmon propagates in such a small, with such a small, let's say, boundary condition or localization, that the plasmon excitation, the plasmon wave, becomes a standing wave with a very simple shape. So basically, for a sphere, it's just a dipole. You just excite the electrons compared to the ions, And at optical frequencies, the electrons are oscillating very quickly, but the ions do not move. They are too heavy. So basically, you have this ball of negative charges moving with respect to the positive charges. So this is just like a giant dipole. So it interacts very strongly with light. It's very easy to observe. So we can do nice experiments with it. And we like to play with it a lot. So another thing we do is we can change by synthesis the shape of the particle from a sphere to a rod, elongated cylinder, if you like. And if we do that, we tune the plasmon resonance. I will not go into the details of why this happens, but it's very convenient because by changing the aspect ratio, by taking more and more elongated particles, we can tune the frequency of the plasmon all the way from the green to the infrared, near infrared, 1 micron. So it's convenient because we can adapt each nanorod to one molecule or to one laser or to one laser and one molecule. So it's a very beautiful Lego play, Lego game. So this is just an example to show you the tunability. These are the scattering spectra of 2 nanorods, and you see that we have a narrow resonance. And as always in physics, when you have a narrow resonance, think of the fluorescence, you have enhancement of the effects if you are exactly at the resonance. You have more amplitude for any optical effect that you would like to measure, and you can also use that to enhance fluorescence. All right, so let me briefly talk about one application of this, which is Stokes and anti-Stokes photoluminescence of gold. So when we started to work with gold, we thought, yeah, gold is nice, it's a metal, but it will absorb a lot of photons, so we can measure absorption. But normally it should not luminesce, right? A metal is not fluorescent because there is no gap. In a molecule, you have electron and hole, they can recombine. In a semiconductor, it's the same case. Electron and hole have minimum gap, they can recombine, get a photon. In a metal, electron and hole will go all the way down and recombine without giving you any photon. This is not completely true. Actually, these 2 guys in my group have been working on that. So Achilles Caratino, who defended his thesis last year, and Martin Caldarola, who is still postdoc in the group, they decided to look at the luminescence spectrum of gold nanorods and to compare that to the scattering spectrum. And you see that for different nanorods, the green spectra, which are photoluminescence, and the blue spectra, which are luminescence, resemble each other very much. They are not identical, but they resemble each other very much. And so the idea came then, why not use photoluminescence, fluorescence, instead of absorption or scattering, because it's more convenient. All our setups are created to detect fluorescence, so we just put a gold nanoparticle instead of putting a molecule, and can we see the photoluminescence or fluorescence? It's called fluorescence for a molecule, but photoluminescence for a gold particle. And the answer is yes, because although the quantum yield of this luminescence is extremely weak, it's on the order of 10 to the minus 6 or 10 to the minus 7, very, very weak. Nevertheless, because there are so many photons absorbed by the particles, quantum yield of 10 to the 6 is quite a comfortable signal to detect, as you can see here. So what Achilles did immediately is look at photoluminescence spectra, not only on the red side of the excitation. This is a laser excitation. You see here, you have a red wing. There is also a blue wing that's anti-Stokes. So this is a bit surprising that you not that you can see it. You note in Raman scattering you have Stokes shifted light as well as anti-Stokes shift light. Both are there. But in the case of the nanoparticle, if you choose carefully the nanoparticle and the laser, you can have this situation where the anti-Stokes is stronger than the Stokes, which is surprising. Why is that? It's just because here we have the plasmon resonance, is the photoluminescence spectrum when we excite at 532 with green light. And you see that the plasmon resonance of this nanorod happens to be at about 600. And therefore, if we coincide the anti-Stokes emission with 600, we get enhancement. Thanks to this resonance, we get enhancement of the emission, whereas there is no enhancement or minor enhancement of the Stokes emission. So that's the way we can tune Stokes versus anti-Stokes. And what is the advantage of seeing anti-Stokes? There are 2 advantages. The first one is we can enhance the contrast. If we have molecules, if we have dyes or flavins or autofluorescent molecules in the cell that will absorb this wavelength, most of the emission will be Stokes shifted to longer waves. There will be almost nothing at shorter waves. It's not completely true. There are hot bands, but these hot bands with dyes are normally very weak. So if you take the following sample, for example, this is a piece of glass with cells that we have grown, and then we have stained these cells heavily with a dye. So in principle, you don't expect to see anything because there is so much dye here, high concentration. Nevertheless, you can, with the anti-stokes, you see in the stokes, you see nothing. Where the circles are, we know there are nanorods, because we have imaged them before. And you see that it's not possible to see anything. Look at the huge fluorescence, 10 to the 5 counts per second. If you go to anti-stokes, the background is much, much lower, 10 to the 3 instead of 10 to the 5. But the nanorods now appear completely on a very low background. So it's possible in this way to decrease the background very much if we have labels that show significant anti-stokes emission. And the second advantage of these anti-stokes is that this wing here, of course, we come with this laser and we go out with a photon that has more energy than the laser. So we need to take energy from somewhere. So this energy comes from the thermal bath. It means we need to extract energy from the molecule here. We need KBT. So this wing here is strongly dependent on the temperature, on KBT. So that's a way to measure this temperature. So that's what we did here. We measure carefully this blue wing. Now it's plotted in energy. So this intensity of this anti-Stokes wing as a function of energy, normalized to the plasmon resonance, and you see you can fit. And from this fit, you deduce the temperature. So this is a nice way to measure the actual temperature of the gold nanoparticle. And that's important because, as you know, nowadays people try to use photothermal therapy, which is attaching gold particles to cancer cell, for example, and heating the gold nanoparticle to kill the cancer cell. That works well, but if you do that, you have to control the temperature very well, because if you heat too much, you not only kill the cancer cells, but all the cells which are around. So you need to know not only the temperature of, you need to know the temperature of the local heating spots very precisely. And this is possible with this technique. So we hope that this may become useful in the future. I have to conclude quickly. So I will just briefly mention the last subject. So when we have started studying this heating, we came upon the question, is it possible to create bubbles, to boil water around a nanoparticle? Because we could calculate that the temperature elevation was really several degrees, or tens of degrees in some cases. So can we boil solvent, in principle water? This is also interesting because if you can do that, you know the boiling water creates nanobubbles, and these nanobubbles are a very strong perturber, mechanical perturber for the environment. You can in this way also kill cells by creating bubbles around the particles. So can we understand this process better? I will cut the story short. There's a work essentially of Lei Hao, and his successor is Thomas Jolans, is now in the group. And I will not show you the details, just a trace. So when we send a CW constant laser on a nanoparticle with enough power, what happens is this very noisy trace in the scattered light. So it means actually that we have bubbles. So these bubbles occur almost periodically when we excite continuously. And it's even possible under certain conditions to detect the sound waves. So that's what you see here. We have the explosion due to the bubble. And then a while later, something like some tens of nanoseconds later, comes an echo. And this echo is just a reflection of a sound wave on the frontals of the objective. So you see that this is possibly a technique that can become useful not only in photoacoustic microscopy, but maybe in photoacoustic therapy. So I will stop here, and thank you for your attention.
Thank you. So I think for sake of time, maybe we limit to one or two short questions while Xiaowei is preparing for her presentation. So are there any questions? Oh, you have to use the microphones.
Yes.
Hello, Rudolf. Good evening.
Hello, Michel. My name is Rudolf Riegler. Coming back to your copper azurine fluctuations and the others, I just recall the history. Sunny C and Peter Liu, colleagues of Xiaowei, have shown that you have the fluctuation in the glucose oxidase. Right. It was apparently a conformational fluctuation.
20
Lars
years
Edman
ago.
and myself, we have shown that for the horseradish oxidase, namely for the making the product, the fluctuation in the product. So the idea is you have a whole manifold of structural conformations, a conformation matrix, and you're shuttled between this conformational matrix. Now, this has been found in several other cases. Lipases and others where the meeting in Leiden, you might remember that some years ago, when it was taken up. Now, you have shown exactly the same. However, you have one case where it doesn't fluctuate at all. It's always the same, this zinc azurine atto system. Why don't you see this fluctuation in this case? This is my question.
Yes. So the question is, what is the difference between the copper azurine, where you see these changes of rate as a function of time, and the zinc azurine? So there are two differences. The first difference is that, of course, zinc is zinc 2+ is a closed shell, so it doesn't like to lose and gain electrons. So there is no redox reaction with zinc. So it doesn't accept. And the second difference is that the dye that we put on the zinc azurine is different. And we did that because we wanted to have a time-dependent signal. In this case, it's redox reactions, not of the metal center, but of the dye itself. So this Atto 655 is different from the Atto 647N. So for the copper, we want to get rid of the dyes reactions. So we took, in this range of potentials, Atto 647N is better. We just took the zinc azurine with the dye to have a time-dependent signal that we can compare. Otherwise, the zinc is completely constant. We also did this control, of course. The zinc azurine has no change of signal because the dye is on all the time. So you see no correlation and no dynamics at all.
But you say that's the sensor pair which you're using that does not show intrinsic fluctuation in the conformation even of the azurine.
Yeah, I'm sure there must be intrinsic conformations, but we cannot detect them. We have no detector. Yeah, we have no electron transfer, so we have no detector of the conformational changes. But they must be there too, yes.
This is to this interesting experiment with in the glycerol, the perilene in the glycerol. And now
yes, so I think in the case of glycerol, where you have this difference from molecule to molecule.
Some molecules move slowly, but the others fast.
So it's not only the molecule that is different, but the molecule and the whole environment of the molecule is different.
So is that due to the solvent?
Yes, yes, indeed, yes, yes.
Okay, I think we thank Michel for this excellent presentation. And for the sake of time, we move on. Hopefully there will be some time also at the end of this session to ask additional questions. So keep your questions till then also. So let me then introduce Professor Xiaowei Zhuang to give a presentation. And I think we will hear about super resolution microscopy and also about RNA imaging. So I look forward. Thank you very much.
Thank you. Thank you, Jerker, for inviting me here. And I would like to congratulate you too for being elected to the Royal Academy of Engineering Sciences. Yes, Jerker said I will talk about super resolution imaging, and which is the first bigger half of the talk. And towards the end, I will also talk about a new imaging approach that we developed, more addressing things at the system scale or genomic scale. And I often like to start my talk with this slide and give you some background or motivation of why we develop these imaging approaches for looking at cellular or biological systems. And as we know that and as also Jerker has introduced, that in the cell, or biology is so complicated, there's so many different kinds of molecules, and in the cell, all these different kinds of molecules interact and really form intricate interaction networks, and then together give the cell its behavior and function. And then we really like to use imaging approaches to study these biological processes. And then with that, then we think the imaging approaches better have these properties. And first is it better has a molecular scale resolution so that we could directly visualize molecular interactions. And then because many of the biomolecules, such as proteins and RNAs and so on, are nanometer in size, then molecular scale resolution means nanometer scale resolution. And as I mentioned, and as Jirka has also mentioned, there's so many different kinds of molecules, so we would like to have molecular identity in our images, meaning molecular specificity. And then we would also like to have dynamic imaging capability because these molecules are constantly changing and moving around and so on in living systems. So with that, then it quickly comes to fluorescence microscopy being a pretty good candidate for getting these properties. And with fluorescence, we already heard, so I don't have to talk about its molecular specificity because we could have multicolor and we could have specific labeling of biomolecules. And these give us the ability to actually look at the specific molecules of interest. And of course, being a light microscopy approach, its relatively low invasiveness allows us to look at live systems. And traditionally, light microscopy wasn't so great on the nanometer scale resolution front or molecular scale resolution front. And we know that is because of the diffraction limit that limits the resolution to about half of the wavelength of light, which is 200 nanometers, which is about 1 to 2 orders of magnitude bigger than the molecular scale resolution that we would like to have. And of course, it's a well-known thing that this resolution barrier has been circumvented already with super resolution imaging approaches. And there's actually multiple different kinds of super resolution images. And on the optical front, there are 2 primary categories. And one is using pattern illumination to get effectively small point spread function to overcome the diffraction limit. And the pioneering approach is the stimulated emission depletion microscopy STED, pioneered by Stefan Hell. And later on, he generalized it to a resolved concept. And then also the structure illumination microscopy in its saturated form also overcome the diffraction limit. And what I will be focusing on is actually single molecule-based super resolution approaches, STORM and PALM, as you have heard from Jirka. And it's really great to follow Michelle's talk on this because we have heard from Jirka, Michelle is really a pioneer in single molecule detection. And in the 1989-90 WE and Michelle demonstrated single molecule detection through absorption and fluorescence respectively. And also I'm really glad to see Rudolf Regular sitting there, who's another pioneer in single molecule detection through his development of FCS. And of course our symposium chair, Jirka, for doing this beautiful FCS work together with Rudolf. And so the thing about single molecule detection, actually, as I like to say, that is it's known in the single molecule field for quite a long while that even though the image of single molecule is diffraction limited, so that the width is 200 nanometer wide, nonetheless, we can determine the center position of the image with just, much better precision. And that precision is the image width, which is the diffraction limit divided by the number of photons that we detect. And if you detect 100 photons, you take a square root, that becomes 10. And if you take 200 nanometer divided by 10, you get 20 nanometer, right? So, but I also like to clarify that this localization precision doesn't really overcome the diffraction limit yet, because the diffraction limit resolution means when objects are too close, they're not resolvable. So when you have two molecules that are separated by distance that's less than 200 nanometers, their images overlap, and then you can no longer resolve that. So the essence of overcoming the diffraction limit is actually this, how to solve this overlapping problem, which in biology could be pretty severe because we're often not talking about 2 molecules, but hundreds or thousands or more molecules that give you overlapping images. So how do you solve this overlapping problem? And that's where we came in, and we and others had come up with the idea of using the time dimension to solve this overlapping problem. Meaning that we do not turn on all the molecules simultaneously, but only a subset at a time. Such a small subset so that their images do not severely overlap, and then we can pinpoint their center position. And then we turn off this subset, turn on a different subset, and pinpoint their center positions. And then we iterate this often thousands or sometimes even tens of thousands of times until we determined all the molecular positions in the field of view, or a sufficient number of molecular positions. And then we can put them together. I'm still trying to learn how to use this cursor. The laser pointer doesn't work. So then we can construct the super resolution image by these molecular positions. And then now you can see that this resolution of the image is no longer limited by diffraction or how wide these images are, but how precise these images, we localize them. And then, as many of you know, that as we were developing STORM, they were parallel and independent work. And for example, that by Eric Betzik in collaboration with Harold Hess and Jennifer Limpinkoff-Schwartz, and independently developed a similar approach called PALM, and published basically simultaneously to our STORM work. Within a week, these 2 papers came out together. And then just a few months later, another lab published a similar technique called FPALM, and that's by Samuel Hess' lab. And then here is the demonstration of how this works, that you activate a subset at a time, you pinpoint their center positions, and then after enough accumulation, you can get this kind of image of a biological structure called microtubules, and this is the comparison in the same field of view of diffraction-limited image, which you can immediately appreciate how much resolution gain that we already have. Then with that, then we rapidly then developed a 3-dimensional, this is a 2D super resolution image, and then since biological structures are 3-dimensional, and we really need to overcome the diffraction limit in all 3D. So then we realized that I skip a lot of basic details to show that we realized that we can determine the XY position from the centroid of these images, but we can determine the Z position from the shape of these images. And when you combine X, Y, Z coordinate, then you can get this kind of a 3D STORM image. And in the first 3D single molecule-based super resolution imaging approaches that we demonstrated in this work, we actually get 10 times better resolution, 20 nanometer in X and 50 nanometer in Z, 10 times better than diffraction limited in all 3 dimensions. And I like to say that that was sort of a point, depending on whether you're a biologist or whether you're a physicist, you have very different view. If you're a or technology developer or application people. My biologists in the lab were very happy with that, so they move on using that to make biology discoveries, because after all, it's 10 times better, you get much clearer images of the cell. My technologists in the lab felt like, wow, you keep telling us it's important for molecular scale resolution. This is still about 10 times off the molecular scale, so there's still room for development. And then I have to really quickly fast forward a lot without going into details to say that we and other labs have been working towards keep improving the resolution. And this is just for the time's sake, a few of our own work where we can use better imaging schemes and better, more brighter probes to actually get to a few nanometer resolution. And then we can also we have also demonstrated live cell STORM with dynamic imaging capability, actually now with sub-second time resolution and 20 nanometer spatial resolution. And then indeed, it's an approach, you know, I have to say, because it inherits the good properties of fluorescence microscopy, that the STORM is widely applicable to a lot of biological systems. And then this slide is a subset of our biological applications, which just demonstrate that we can apply it to a variety of systems that range from relatively simple cellular systems, such as bacterial cells, you know, sperm tails, mammalian cells, and so on, all the way to complex tissue systems where we actually do not have to do monolayers of cells. And the storm is capable of going relatively even deep into the tissue to get tissue scale biology to look at, in this particular case, subsynaptic structure and neural circuits. But I would like to actually just focus on one biological application to demonstrate the power of super resolution. Because I often found it was very proud for me to say that super resolution can not only be used to get prettier, sharper images and then improve the understanding of known biological structures, but actually can be used to discover biological structures that we didn't know existed before. And that came from our imaging of actin inside the cell. And actin is a very well-known molecule to be very important for a variety of cellular properties, such as the cell mechanics, the cell shape, the cell biogenesis of many organelles inside the cell, the transport, and so on. But what people typically know about actin structure is this kind of actin structure. This actually was the first, this was a storm image. It actually is the first optical image that you can actually resolve individual actin filaments inside the cell. We were pretty proud to get that kind of structure, but I also like to tell people that through this, we really didn't learn new biology because it all came to be just the same as what people have done electron microscopy and have already demonstrated that these are filament form crisscross network type of structure. But what was really surprising and a true discovery of things that didn't know before, really surprised everybody, is when we looked into axons for what actin looked like. And you can see this periodic sort of stripes that you can see in the axons. And as I said, it was very surprising to people because the high-resolution imaging of neurons has existed for 50 years, and then people don't know how they just missed the structure. And optics is not hard to explain because if you turn on just the diffraction-limited optics, you completely miss the structure because it's just a smear. And I'll show you the dimension of the structure. It is sub-diffraction limit. And but if you're curious about why electron microscopy missed the structure, I don't have time to talk about it during my talk, but if you ask me that question, I'll tell you why electron microscopy missed it. So going back to the structure, and it is, so this is a 3D storm image. The color indicates Z, which means we can actually take any of these stripes, turn around, and these are rings. So it's a really interesting ring-like structure that wrap around the circumference of the axon underneath the axonal membrane. And a very important property of it is its periodicity or its regularity, regular spacing, which you can actually do the Fourier transform or autocorrelation of the axon to see it truly is a highly periodic structure with a period that is 180 nanometer, which is smaller than diffraction limit, which explains why diffraction limited optics give you a complete smear. And the 180 nanometer is also important in inspiring us to learn more about this structure. Because when we look at it, we couldn't help just thinking that these actin rings must be spaced by a molecular spacer. And this molecular spacer must be 180 nanometer long. So I'm just speaking up the obvious. And another obvious thing is it must interact with actin. These clues actually led us to this molecule called spectrin, which is actually well known in an entirely different cell, the red blood cell. And underneath the red blood cell membrane is a skeletal structure that looks like this. And actually, I would like to say that this red blood cell structure has still yet to be seen in this structure in an intact red blood cell. But if you isolate the membrane out, and then you can use EM to actually see this hexagonal kind of a lattice structure. And then these arms are just the spectrin tetramers that are about 180 nanometer long. You know, you can see that's one of the reasons why I think of it. Another reason is that these nodes of these lattice is actually this green cylinder is a short-acting filament that directly interact with spectrin. So that, because of that, we think the spectrum is a good candidate for this spacer. And we came up with this hypothesis of our newly discovered structure, which is a short-acting filament aligned into a ring structure. And then they're periodically spaced along axon because they're connected by these 180 nanometer long spectrum molecules. And of course, so we need to test to show that's true. And to test this hypothesis, we did two-color storm. And we detected in various ways different components of our hypothetical structure. We did two-color storm images of actin and spectrin. And better than spectrin, we actually were able to detect a specific location of the spectrin tetramer, which is the center location, which we expect red, green, magenta alternating rings. And that's exactly what we detected, green, magenta alternating stripes. And then we also think that if it's a short-acting filament, it should be capped. And then we actually discovered the capping proteins in the structure too, or identified it. And that's one end is capped by adenosine. And indeed, you can see adenosine when you image it together with the center structure, center position of the spectrum. It's also alternating. And we also found the other capper, and that is triple modulin. And we also found that the structure has many other proteins. And one of them is quite important, called anchoring, because it actually is an adapter that anchor important membrane proteins onto the structure into a periodic distribution. And one of them is sodium channel in the axon initial segment, also has the periodic distribution. And the sodium channel, the ions flow through sodium channel is responsible for action potential generation, this electric pulse generation in the neuron. So we think this structure is actually important for action potential generation. And since then we have discovered many molecules on this structure, many important, functional membrane molecules included, but I won't have time to go into that. And then I want to also say that subsequent to this original discovery work here in our lab and also independently Stefan Hell's lab have shown that the structure also exists in dendrites. But in dendrites, it tend to not form with the same propensity, meaning that not the entire dendrite would have this structure. Also, it forms much slower in dendrite than in axons. So in dendrite, it has a lower propensity and slower development rate. And then since then, we found even more surprises for this structure, and that is both in some regions of dendrite and in the cell body, we actually discovered this two-dimensional hexagonal lattice structure, and which actually resembles that of the red blood cell structure. So, so now we have a more complete picture of this newly discovered structure, which we named it MPS, membrane-associated periodic skeleton, which has a really interesting story behind such boring name, which, yeah, I actually won't go into that because of time limit. So let me just tell you that this is now our relatively comprehensive understanding of the MPS structure in neurons, where it forms one-dimensional periodic ring kind of a structure in axons, and then also in a substantial fraction of dendrites. And then it forms the two-dimensional periodic, two-dimensional lattice, polygonal lattice structure in the cell body, and in a small fraction of the dendrites. And this is really a ubiquitous structure that we found that it exists in all kinds of neuronal types, excitatory, inhibitory, peripheral neurons, central neurons, myelinated, unmyelinated. And also in 2016, Stefan's lab has an independent publication showing this structure in a overlapping but different subset of neurons. So basically, we both think that this is a structure that exists in all neuronal types. And then we also demonstrated that this structure is a highly conserved across species. At least we found it to be starting from C. elegans all the way to humans, the structure all exists. So with that, it really indicates that it has to be an important functional structure. And there are multiple functional roles that we still keep discovering, but I want to tell you a few of its functions that are known, that we already know. And then one is mechanical. And I often show this. And I want to save time for my RNA talk, but I couldn't resist. I always demo that. And this was actually a toy from my daughter, so I thank her for giving me this toy. So with this, you can already see what do I mean. It's a structure that gives you, because it's a rigid acting ring linked by flexible spectrum, so it gives you this bendability or elasticity while still keep the shape intact. And it is what I like to say that what the axon really needs, because it's really long, really thin, and under mechanical stress. And then there is indeed experimental evidence for that, because if you knock out spectrum from C. elegans, you will see axon tend to spontaneously break. And not only that, it only breaks when C. elegans move. If C. elegans don't move, the structure can't, the axon can grow to normal shape or morphology. And then because it anchors important membrane proteins, as we demonstrated, it also is important for a variety of axonal signaling pathways. And then the rest I'll go fast. It's also important for the formation of some of the very important axonal structures, such as the axon initial segment and nodes of Ranvier, where the electric pulse is generated and amplified. And it's important for the morphology of axons and dendrites. And importantly, if you disrupt the structure, and there's true medical consequences, because you can generate widespread neurodegeneration and a variety of neurological impairment. So with that, let me just quickly go to the next part, which I already prepared a shorter part in case Jirker is nervous about timing. So, but I do want to get the motivation very clear, okay? So because this is my new excitement, and we realized that inside the cell, we don't just talk about 2 kinds of molecules or 3 kinds of molecules. There really is thousands of different kinds of molecules that work synergistically, collectively, give the cell's behavior. And we also know, as imaging people, that imaging usually have a low throughput when it comes to imaging molecules, because we typically use 2-color or 3-color images to study 2 or 3 kinds of molecules. But we really want to have the genomic scale throughput to be able to probe all these molecules simultaneously to give us a system scale understanding of the cell. So we want to add this important property to imaging. And then I want to demonstrate, we're doing it for both RNA and DNA, and also are interested in protein. But let me actually just demonstrate it with RNA. And the reason for doing that, because some of you might know that a genomic scale RNA detection is possible by sequencing, right? So single cell RNA-seq, which has begun to sort of revolutionize many areas of biology. So then why do we actually want to develop an imaging-based approach? And there are a variety of reasons, and in situ is really the major reason. And that gives us 2 properties. One is we do not have to dissociate cells. We can directly study intact tissues, and then you don't introduce perturbation. And the second is because you do it in situ, you get an image, you actually can get directly the spatial information. And spatial information are important at 2 levels at least. One is inside the cell RNAs are not uniformly distributed, and local distribution of RNA are actually important for a variety of cellular functions. And also at this tissue level, cells are not uniformly distributed, and this I want to emphasize a little bit more. So because of the spatial organization of cell is truly important for tissue function, and then if we could actually, without dissociating the cell, like in RNA-seq, and actually use our imaging approach to do transcriptome profiling of individual cell and then identify the cell, then we can get the spatial organization of the cells, different types of cells, directly. And the approach that we use is a single molecule FISH-based approach. And single molecule FISH was originally developed by Rob Singer, which you use counter oligos to actually fluorescently label to identify RNAs inside the cell so that you can image individual RNA molecules and count their number. But the problem about it is, again, just the imaging-based approach. You can image 2 or 3 kinds of RNA. But if we're talking about transcriptome scale, that means thousands of RNA. So how do we get these many orders of magnitude to get to the genomic scale? So that, so a few years ago, I came up with this idea of using combinatorial labeling, sequential imaging, plus error-robust barcoding. So here's the basic idea. We barcode each RNA, or we encode each RNA with a binary barcode. And then the different RNAs have different barcode, like 1010 and so on. And then we imprint this barcode on RNA by hybridizing oligos. And these oligos can not only bind RNA, but they have additional sequences that are determined based on this barcode, meaning that if it's 1010, it means I will have a readout sequence 1 and readout sequence 3, but not other readout sequences. And then I determine which readout sequences are on the RNA by additional sequential rounds of imaging. In the first round, I only read out readout sequence 1, means I only detect those RNAs. Their first bit reads 1, but not 0. In the second round, I only detect those RNAs. Second round reads out 1, but not 0. So after n rounds of imaging, I get individual RNA molecules inside the cell. Each dot is one RNA molecule, and then each one of these RNA molecule has a binary string barcode attached to that. And this is very powerful because after n rounds of imaging, I can distinguish 2 to the n different kinds of molecules. And then if n is equal to 16, 2 to the n is 65,000, and that's actually the transcriptome size. Here I just go through very quickly that because each bit has an error, finite error in it, so we actually develop error correcting barcodes to actually get us to have a very high accuracy of this kind of approach. That's why we termed our approach multiplexed error robust fish or MERFISH. And then these are just a couple of slides to show that it works beautifully. So this is at the 100 gene detection level, we can actually compare our MERFISH detection with if you do it one at a time with single molecule fish. My student only had the patients to do 15 of the 140 that we detected, but of the all 15, they agree perfectly in terms of abundance. And then we can actually compare all 150 with bulk sequencing to see high correlation. And with that, we get a detection efficiency of nearly 100% with very low, actually less than 5% misidentification rate. And then we can also detect 1,000 molecules, and then again still get high accuracy and high detection efficiency. So, so this actually represents the first demonstration of the transcriptome scale single-cell RNA profiling by multiplexed fish. And there are other labs who also appreciate this kind of imaging-based approach for transcriptome scale study, but using different approaches. For example, Mats Nielsen, right here in Sweden, and George Church, they develop in situ sequencing approaches to do this kind of thing. So in terms of application, I would only have time to show you one, and I felt like I set it up that way, then I really want to demonstrate we could do that. Remember what I said, that being an in situ approach, I don't have to dissociate the cell from the tissue, so I could actually transcriptome profiling individual cells directly in tissue based on the expression level. And I should say that the reason that there are different kinds of cells, their different behavior and function, is not because they have different genome. We all know that these cells have nearly identical genome. It is their transcription profile that's different, that caused different cell type or different behavior and function. And because we can quantify this transcriptome of individual cells, then we can in situ identify cell type, and then we can get their spatial organization. And then here's a demonstration in the brain region. We can actually get different cell types. And in this brain region, there are actually nearly 100 different cell types. I only displayed 10 here. And then you can see their truly interesting spatial organization that actually changes as you move from one brain region to another. I think I will skip that part. And just with that, I want to end always by this slide to show that with different imaging approaches that both our lab and other lab developed, we hope that someday, this is not reality yet, we hope that someday we can have this kind of images of what's happening inside the cell so that we can see molecular events unraveling right in front of our eyes like a movie. So with that, I want to just quickly thank the people who has done the work and the MERFISH development, some pioneering students and postdoc in the lab, Mike Rust, Mark Bates, and Bo Huang, with some other generations of students. And the periodic structure, Ke Xu, Guishen Zhong, and more recently, Jiang He, Boran Han, and Robo Zhou. And the MERFISH development, really, Jeff Moffitt played an instrumental role together with Alistair Battiger, Kakao Chen, and more recent generation of people. And the brain imaging of MERFISH is really great collaboration with my long-term friend and colleague Catherine Dulack's lab at Harvard, and the work is supported by Hughes and NIH, and thank you very much for your attention.
Thank you very much, Xiaowei, for an excellent talk.
Sorry for those people who heard the longer version an hour ago. I didn't know there were overlapping audiences.
I think it's not that much of an overlap. They don't mind.
Okay, good.
So is there a short question while we are switching speakers? No question? So sorry.
Hello?
Yes. I know who you are. Yes.
Because, I mean, the RNA molecule, or it's too big, or you think it could be used in any way?
So no, actually super resolution would be wonderful. So there are two things to think about it. The MERFISH image itself is already a super resolution image, even if each round of hybridization we do a conventional imaging. Because the image come out of 16 rounds, or 4 rounds with 4 colors, then originally would be overlapping images because molecules, we actually resolve them. So in that sense, it's already a super resolution image. It's just in this particular case, we activate is a quotation mark, molecules by knowing their species, right? Rather than the stochastic activation as we did in STORM. But that said, there is still, useful. It's still useful if we want to do super resolution imaging in each round. And the reason is because when we want to detect a lot of molecules, say 1,000 or eventually 5,000, 10,000 molecules, then the neighboring molecules can get really close. And then there, even within each round, one of the 16 rounds, you would start to have molecular overlapping problem. And there, having super resolution like STED and STORM, it's going to be very helpful. We actually already demonstrated that with another super resolution approach that I didn't have time to talk about, and that's not an optical super resolution approach, as you probably know, the expansion microscopy, right? You expand the cell, and then you can get effectively super resolution with diffraction limited optics. So in some cases, when we have high density of RNA, we found that that approach, actually is pretty helpful for resolving neighboring molecules.
Okay, thank you very much. So then I will give a short presentation of some of the research activities in our research group at KTH. So we work with, ultra-sensitive, ultra-high resolution fluorescence techniques. We have heard about fluorescence correlation spectroscopy and single molecule spectroscopy. I will not talk so much about that, but rather concentrate on STED microscopy and trust microscopy, focusing on how these methodologies can be used towards clinical diagnostic applications. So STED microscopy we started with almost 10 years ago by now. So we did an EU project in collaboration with Stefan Hell and his research group in Göttingen, and they helped us at that time to build up a STED instrument with dual color, and which we then used within this EU project, where we wanted to find new strategies for early detection of breast and prostate cancer. So one issue with these cancer forms is that you do sampling, and preferentially you would like to do sampling with small needles in order to minimize side effects, including risk of spread of cancer. But this means also you have a lower amount of sample, and that can be sometimes a diagnostic problem. So the question in this EU project was, can we compensate for the small amount of diagnostic material, small number of cells, by actually extracting out a maximum information from the sparse number of cells, and using additional sources of information than traditionally used, that you look, for instance, into tumor marker proteins, if there are many of them or less of them and so on, the content, but also actually how the proteins are spatially distributed within cells. And then it's known from biophysical studies that cancer cells tend to have a different mechanical properties compared to normal cells. And we hypothesized that we could then maybe also see that from the distribution patterns of cytoskeletal proteins, and compare, say, the thickness of fibers, the directionality of fibers within cancer cells and normal cells. And indeed, we can see that there is a difference, and that this could form a basis for diagnostics. We also looked into various other features, including membrane protein distributions. So we actually took this kind of concepts also further and applied it into other areas. So this is a collaboration together with the group of Birgitta Enriquez Normark at Karolinska Institute, looking into bacteria and pneumococci. And for these bacteria, they have surface proteins, and of course it matters the expression levels, but also how they are distributed on the surface. Influences how their capability to invade into tissues, to pass, say, the blood-brain barrier, also their susceptibility to antibiotics, and for their ability to evade immune system. It all depends on the distribution of some central surface proteins on these bacteria, which then can be very well quantified and characterized with super resolution STED imaging. Another application area is platelet studies, a work together with Professor Gert Auer and others at Karolinska Institute. And the background here is that platelets are involved in early tumor development, and it has been known for more than 100 years that cancer patients tend to display disturbances in their coagulation. So often cancer patients have repetitive thrombosis in the lungs or in their legs, which has been attributed to a disturbed platelet function. And then later one has found that also protein contents within these platelets actually are different in cancer patients compared to normal healthy persons. And what one believes is that platelets actually that they have an interplay with the tumor cells, such that they can selectively release proteins, for instance, promoting the growth of blood vessels around the tumors, and tumors can induce that. And then rises the question, how can these platelets actually so specifically release one protein but not the other? And given that they are quite primitive cells. So we could investigate that quite well with STED, labeling proteins of interest and see how they are distributed in cells. We can see that actually they are stored in small vesicles, and also that they tend not to actually be co-localized with each other. If you have, say, a pro-angiogenic and an anti-angiogenic protein, they are stored in different compartments, giving a basis for also selective storage and release of proteins. We also studied how these platelets and the distribution patterns varied with activation of say, thrombin or ADP, and we can see that actually the distribution patterns are different for different proteins and for different types of activation. We have also co-cultured platelets together with tumor cells, and we can see that tumor cells actually in a non-contact manner can influence the platelets and change the distribution patterns, such that we can actually identify platelets co-cultured with tumor cells compared to platelets co-cultured with normal cells or with no cells at all. So indicating that we can get hold of the mechanisms of protein release and how that is related to tumor disease. So the other method I shortly want to mention is thrust imaging, transient state imaging. This is a method we have developed ourselves in my group, and one can say as a basis for this that in fluorescence microscopy or spectroscopy in general, you often want to use multiple parameter readouts. Either it's a matter of array readouts, microscopy, flow cytometry. And then you take benefit of the intensity of fluorescence, lifetime, polarization, and emission wavelengths. But what you also can actually use is the blinking properties of fluorescent molecules. We have heard about their use in super resolution microscopy, and also that it's actually limitation if you want to do single molecule microscopy or spectroscopy, but they also represent a information source. So they are extremely environment sensitive. So the blinking properties depend on the environment around the fluorophores. And one way actually to look on that in a very straightforward manner is to apply a modulated excitation and then use some kind of microscopic readout. It can be a wide field microscope, it can be a total internal reflection, it can also be a laser scanning confocal microscope. And then depending on how you modulate your excitation source, you can populate the dark states differently depending on say the pulse width of the excitation pulses, the separation between them, the height, the shape, and so on. So doing that in a systematic manner, you populate the dark states also differently. And it means the remaining bright fluorophores stay fluorescent, and actually the fluorescence also varies then depending on how you modulate. And then depending on how the average fluorescence intensity varies with your modulation characteristics, that information then gives you the information about the blinking kinetics or the rate constants, in this case, to and from a triplet state. And that you can directly then, by recording multiple images with different modulation put on the excitation, you can crank out that pixel and pixel-wise. So in that way, you can generate images, not only, so the upper two ones are normal fluorescence images of cells, the lower ones are actually cells where you have then used thrust imaging showing the triplet decay rate. And this decay rate depends on almost exclusively on the local oxygen concentration. And then we can see that to the left, we have cells showing a high rate, indicating a high oxygen content, a lower oxygen consumption in the cell. On the right-hand side, we have cells with a low rate of the activation of the dark triplet state, indicating a low oxygen concentration and a high consumption of oxygen. And this difference in oxygen actually shows the difference between a tumor cell specific metabolism compared to a normal cell specific metabolism where you consume more oxygen. So it means with this kind of imaging method, we can actually resolve different metabolic types of cells in a manner that is not possible by any other fluorescence parameters. And we can also apply it to a flow cytometry system such that we can actually record a larger number of cells and get statistics and actually do flow cytometry extensively used in the clinics using traditional fluorescence parameters. We argue that we can also add to that actually these kind of blinking parameters representing metabolic features of cells, which often reflect different diseases like cancer or bacterial infection and so on. So with that, I stop. I just want to of course, acknowledge my research group. I think many of them are sitting up there in the corner, and don't hesitate to talk to them afterwards also. And thank you for your attention. And I think we move on directly, actually, for sake of time. And let me now introduce Johan Strömqvist. You have his name, or you had his name there. He's one of my former PhD students, one of several brilliant students. And he is now CEO of a company with some 15, 20 people, developing fluorescence methods for DNA sequencing. So please.
Okay, so will it start? Yeah. Hi, congrats for your membership. So my name is Johan Strömqvist, and we are doing DNA sequencing at Singer Technologies, or actually technologies for DNA sequencing. And our ambition is to make DNA sequencing a lot more cheaper and faster. And you might ask yourself, because you probably know the history of the Hugo project, cost a lot of money, and after decades of development, we're down at maybe $500 per genome in the Novasec system from Illumina. And is there a motivation to lower the price even more? Yes, there is, of course. And one of them is, for example, you know this NIPS test, which you do for pregnant mothers. You can detect DNA from the fetus in the blood of the mother and see genetic effects from the baby. Similar stuff is now happening, especially in California, I would say, probably also in Europe, but there's a lot of companies like Grail that want to take a blood sample and they want to detect DNA from potentially a tumor that has leaked out of the tumor. So it's a similar concept. The problem is that there's just a tiny amount for in a cancer patient, especially in early stage, where you can detect this kind of DNA. So for each cancer test, blood test, this company will cost like a whole car. So that's why we have to lower the cost to be able to bring these kind of technologies out. Another thing is the Chan Zuckerberg Foundation, where they want to identify every cell in the body and see how they are localized in a kind of an atlas, and also a lot of development and one of the tech to identify all of these different cell types, as you mentioned, is using single cell sequencing. So suddenly you want to sequence populations of cells, and now we're talking trillions of cells for a human. So yes, we need to lower the price, and that's where we come in. So what has DNA sequencing anything to do with fluorescence? Well, I would say that most of the sequences out there right now are using fluorescence, as you probably know. And single molecule detection, well, you need to distinguish. So typically when you do DNA sequencing, you fragment the DNA and you localize it over a large surface. You take images in cycles, change bases, nucleotides. But you have to be able to distinguish noise from signal. So fluorescence is a very neat method to achieve this. And of course, if you have high resolution, as typically you have in fluorescence imaging, you can then pack the information more densely and thereby make the sequencing of higher throughput. And of course, imaging speed is also very crucial. So I would say that you need all of these three components to be able to do a fast sequencer. You need good resolution, high sensitivity, and very fast speed, which is kind of, well, we call it the eternal triangle of imaging. It's hard to achieve all of these. We kind of push this to the limits with a new tech where we are able to have single molecule sensitivity, confocal resolution, and a speed that is, like 150 confocal system in one machine, basically. So we developed, in our sequencer, a new type of scanner, and also fluidics. So it's not, for DNA sequencing, it's not only imaging capability that is important, you also need fluidics. So that was one thing that was important for the development of fast sequencing was the microfluidic chips, where you could basically use less reagents and speed up the chemistry by confining the the volumes. The last part of the sequencing is the sequencing chemistry. I will not talk so much about that, but typically it's labeled nucleotide or some kind of sensors. So I just want to show some image here of that we have taken on a brain section, thin brain section with our tech. Could you make it big and maybe? So we take 80 square centimeter areas, and we do confocal imaging over the whole area in one without stitching or anything. And we can basically, well, we can basically do the same thing as Google Earth in just one run. It's actually 10 times the scale from macro to micro is actually 10 times better than Google Earth, from Earth to your villa. So we take those huge images, single molecule sensitivity, and confocal resolution. This is actually together with Professor Mats Nilsson at SciLife for in situ sequencing, which is this alternative way of sequence barcodes and identify different mRNAs. Let's see here. This works. So we have this extremely fast scanner, which allows us to make really, really high throughput sequencing, and also use it for in situ sequencing, for example. The other part that we have solved, unfortunately, I cannot go into details of how we do it, but is the fluidics. So typically in a microfluidic system, you have 100 micron thick channels. It's very difficult to make them more narrow. It's a lot of forces shows up then. So what we have done over our samples, we can create very, very thin liquid layers while we're imaging. And these layers can be down to like just a couple of microns. So thereby we can reduce the whole volumes of reagents and speed up the chemistry. So we can both shorten the duration and also make the cycles go really, really, really fast. And this will change DNA sequencing. It will enter a new level when this is commercialized, so to speak. Another part, I think, what is really interesting, I think things are changing now. We focused a lot here during the talk of getting better and better resolution, but and you were talking about the complexity of biology. But what is happening, especially in California, probably also in Boston, is the combination of software and life science or biology. So big data is something that we have to live with, I think, or have to live with. We have to embrace it because, well, the Grail company, for example, the Google has went to that project, and basically you want to sequence a lot and identify patterns from hundreds of thousands of millions of patients, and this kind of amount of data is not a human being's work. It's a computer that should see those patterns and draw conclusions. And I hope we will see more of AI in our industry too. Finally, our niche as a company is actually to be that data generator for big data, and that's what I want to say. Thank you. Yeah.
So I think you indicate an interesting development. May I maybe ask Xiaowei and Michelle to come up, and we take some kind of common question round. I mean, generally speaking, what is the main challenges for fluorescence-based imaging and spectroscopy? Is it to handle this huge amount of data, or what would you say, Michel?
I think it is a big challenge. We don't even start to address it in our group. So we work on mostly with single channel detectors, rarely with CCD cameras. So we are more on the single molecule, one at a time.
Right.
Yeah. So, but indeed, of course, for applications, this is a huge challenge.
Yeah.
Yeah.
So are we. I mean, we don't have that kind of capacity to handle that huge amount of data. I don't know what is your opinion?
Yeah, I think we do, and we do have both the requirement and are working on that. You know, especially on the MERFISH front. You know, we are having the ambition to not only have the very high throughput in terms of how many genes we image, but also, you know, like the whole brain, and then eventually the you know, the human cell atlas and so on, we're part of too. And that is incredible amount of data, and both in terms of hardware storage, transfer of data, and then eventually the most important and tricky part is to analyze such a large volume of data to extract useful information and so on. And so big data handling and processing and information extraction is a very important thing for us, actually.
It's here to stay, definitely, yes.
Yeah. Yeah.
So what do you think? Is fluorescence spectroscopy imaging at its start, or will we see is the boost or the main activities still ahead of us, the applications ahead of us?
Well, you know, I think with all these wonderful development, to say that it's at the start is maybe, you know, not respecting all the things that has already happened. But I think it's or not enough respect the things that's already happened. But one thing that I think you're for sure right is that there are many more things to come. You know, because we're getting into a lot of additional interesting developments that are really just, you know, every day almost you see new things. You know, we are very interested in the genomic scale imaging, but then there are other wonderful development of, you know, clearing technology that lets you see the whole brain by clarity, right? And other clearing techniques and so on. And then there are, you know, ever improving speed of looking at brain activity of you know, animals, so that you can, because there you really talk about, you not only want to get large volume imaging, but you want to get them really fast so that you can record neuronal activities and so on, right? With optogenetics, and both in terms of manipulation and detection. So I mean, these are just a couple of examples. So I would definitely say you're right in the sense that maybe we're not at the start, but we're still very young with lots and lots of great things to come.
Right. I fully agree. And now we have talked rather much about the techniques, but as you indicate also the sample preparations, the fluorophores are very crucial, I think, for the capacity, the performance of fluorescence imaging and spectroscopy. So do you have some questions in the audience?
It's a question for you regarding Storm and Palm, and I think this has been done. So my question is whether it became used, and that is that we have when developing tools to probe molecular interactions, like antibodies, we have aimed for strong binders usually, but there are lots of very weak binders in nature. And you could, in principle, do Palm and Storm by probing transient interactions and track the surface of a cell, or also features within a cell if you can have local illumination. And I remember a paper 5-6 years ago that did something very similar. It was a paper from ETH. And I just found that a very nice approach to super resolution imaging, and I wonder whether that has taken off, or if you see a potential in utilizing weak interactions to fairly rapidly probe structures by using various types of very specific but transient binders for the purpose of imaging.
So are you asking for using these weak interactions for detection, or are you asking for detecting weak interactions?
For imaging. So you just
you
imagine
want to
the
image weak interactions inside the cell?
Image a cell by utilizing weak interactions. So you have the image of a cell, you have the surface of a cell with lots of structures, and then you have probes that bind specifically to certain regions, but they do that very transiently. So in one image you see exactly what you see in palm and storm. You have them fluorescently labeled. And I was impressed by that paper when it came, and then I thought this could be an alternative, and I wonder how if it has developed or whether you see a potential in that.
So the reason I asked that is I think both directions are very interesting. Okay, so using transient binding as a way, as if it's like the stochastic activation, but you have sort of a stochastic binding, and then because of that, you could also separate signal over time, and then you can use that get super resolution imaging approach. And then actually shortly after the publication of Storm Palm is the publication of Paint by Robin Hochstrasser, and then later on there are the further development of Paint by Daniel Choquet's lab, and then by Punying's lab, using DNA and so on. These are all using transient interactions to get these single molecule binding events, and then you can accumulate over time, get super resolution imaging. So that certainly is, a pretty useful approach, I would say. And but the reason I asked this other thing is that there are a lot of transient interactions that are happening inside the cell. And these are the events that are harder to study than strong binding events. And then having high resolution, both in terms of time and in terms of space, is pretty good for detecting these transient interactions. And then there you could think of the kind of approaches like earlier, like the single particle tracking Storm and Palm, where you turn on a subset of molecules and then just track them over time, and then you can see transient interactions there. And then there, the Storm and Palm thing is actually overcoming the density limit, because if they're too dense, they're all happening, you couldn't see these interactions. But if you activate a subset at a time, then you can track and then see these weakened interactions. And that actually leads me to another really very impressive new development of super resolution imaging is this mean flux method that Stefan Hell recently published. And there it's sort of a combination of the Storm Palm kind of thing, that you use stochastic activation to separate the molecules in time. And then you use a pattern illumination like the donut beam to actually have much higher localization precision at a low photon flux, right? So with that, they were able to get sub-millisecond time resolution. We're talking about not milliseconds, sub-millisecond. Then you can see these transient, and also not only that, another thing is using low photon flux, you can get many, many time points. So with that, this very weak transient interactions in the biology that used to be difficult to detect, by methods like mean flux, you would be able to detect that. So both using it as imaging and detecting the interesting weak interactions in biology are happening, and it's really exciting.
So thank you very much. I think for sake of time, we will have to wrap up, and let me, on behalf of the Swedish Academy of Engineering Science, hand over a small gift. And thank you very much. And also for Johan.
Oh, thank you very much.
So thank you very much.
Thank you.
And now I think for those of you who have registered for dinner will be served out there, I think. Right.